Good evening, everyone, and welcome to another screencast in Immunology and Serology. I'm Dr. Supachi Basit, your lecturer for this session. And for today's session, we will be talking about the transplantation immunology. For this particular topic, we shall be discussing the histo. We will be actually reviewing the histocompatibility system because this have been our discussion for during the introduction in immunology. What are the different types of grafts? The mechanisms of graft rejections and graft-versus-host disease, and some people who would be who have received um, transplanted organs may be asked to drink immunosuppressive agents. What are the different laboratory methods to type HLA antigens and eventually identify HLA antibodies? And what are the different molecular methods for determining HLA alleles? Okay, so let's review your major histocompatibility complex, or the MHC. So as we all know, this is actually closely linked genes found on chromosome number 6. And these particular genes or MHC is actually highly polymorphic, thereby can induce a very strong graft rejection. So there are actually two types of proteins. We have the class 1 proteins, identified as HLA, B, and C. Incidentally, all class 1 proteins are expressed on all nucleated cells, including kidneys, liver, okay, what have you. And then cluster proteins are HLA-D. So examples are DR, DQ, and DP. So HLA-D, okay, proteins are actually expressed on antigen-presenting cells such as macrophages, dendritic cells, mast cells. Okay. So other systems are also involved in graft rejections, such as the minor histocompatibility antigens, the MICA antigens, the ABO blood group antigens, and the KIRs. Okay, the MICA antigen is also known as the major histocompatibility complex class 1 chain-related A molecules, or may I say it again, the major histocompatibility complex class 1 chain-related A antigens or molecules. And then the KIR, so KIR stands for the killer immunoglobulin-like receptors. Okay, so these are considered as other systems that could be involved or implicated in graft rejection. Okay, so moving on, we have here an example of the possible progeny, okay, when polymorphic genes are actually combined with one another. So how HLA haplotypes are being inherited. So the thing that these genes that you can see here are the paternal genes, and the genes that you can see here are the maternal genes. So always remember that HLA genes are actually linked and inherited in the classical Mendelian fashion as haplotypes. Okay, so remember your lessons in cytogenetics. So one paternal here and one maternal haplotype could be passed on each offspring. Okay, so 4 different combinations of haplotypes are possible in offspring. So elucidation of haplotype sharing between siblings is an important assessment whenever you are searching for a transplant donor. So for example, this sibling A, the first sibling got this haplotype from the father and this haplotype from the mother. So there are 4 possible different combinations. So if AB comes from the father and then CD comes from the mother, so possible combinations are AC, AD, BC, BD. So it means that even if you are sisters, brothers, your siblings in general, there is no possibility. There is still a possibility that you may not have the same or inherit the same haplotype. Unlike if you have twins, because the haplotypes, okay, would be the same. You'll be inheriting the same haplotypes, each from the mother and from your father. Okay. So, however, instead of getting random organ donor, it is much better if we will be getting donors from our siblings if they are qualified, because there are several screening to be done in order for you to be qualified as organ donor, because the chances of you getting the same haplotypes would be most likely as compared to random individuals. Okay, so that's the reason why we can classify grafts into several types. Okay, so for example, when we say tissue graft, it is the tissue transfer in the same individual, meaning the graft that you will get from one that you will be using from the graft that you will be using in one part of the body actually came from another part of your body. So for example, if you will be having cardiac bypass, you'll be needing a blood vessel to do such a thing. So some doctor would be getting saphenous vein, and that vein will be used for the cardiac bypass. Now, when we say syngeneic or isograft, it is the transfer of cells or tissues to a genetically identical individual, meaning you can transplant between identical twins. Now, allograft is the most common type of grafts, okay? So it is the graft between genetically non-identical individuals but of the same species. So this is the most common type of graft, especially when you are getting organ from your siblings, or even from unrelated donor, from a random donor, or even from an unrelated cadaver donor. Okay, xenograft, however, has never been proven to be effective yet. So it is a transplant between members of different species. Okay, so for example, you'll be getting transplant of pig bulb into human heart. Wow. So there has never been any proof of there has been no documented proof of successful xenograft. Okay, so moving further, there are two types of allorecognition. Okay, it could be direct allorecognition and indirect allorecognition. So what does it mean? Since organs that will be transferred into our body, okay, may not of the same haplotype, for example, in cases of homograft, okay, that particular organ may be considered as non-self by our immune system. Therefore, there are two possible allorecognition that could take place. So one of them is called the direct allorecognition, meaning cytotoxic T cells from the recipient bind directly to foreign HLA antigens on cells of the allograft and eventually release the cytotoxic factors. That way, the organ that has been newly transplanted to you will be rejected. On the other hand, when we say indirect, the host antigen presenting cells coming from the host, okay, the APCs coming from the host, okay, present foreign MHC antigens on graft cells to recipient TH cells. Okay, so direct mainly involve the MHC class 1. Okay, the indirect will involve MHC class 2 because he'll have to do antigen processing. Okay, so these are the two types of allorecognition. So as you can see here, the host cytotoxic T cell, so you can see this is the graft cell. This is the cell coming from your donor. Okay, your CD8 will eventually bind with the of course the T receptor cell here, the TCR, will bind with the CD8. Okay, so what will happen? What will happen is that the host cytotoxic T cell will destroy the foreign cell. First, perforin, which will create pore, and then granzyme, and this will cause apoptosis, meaning programmed cell death of the donor kidney cell, for example, if it's a kidney. Whereas, when you say indirect, antigen-presenting cells such as macrophage, okay, will actually present display. The Graft MHC. These were ingested by the macrophage or phagocytized by macrophage, and then this will be displayed in MHC class 2, and then the T helper cell has a TCR, and it will recognize the CD4, will recognize the MHC class 2. So eventually the T helper cell will produce cytokines, and these cytokines will also destroy the newly transplanted organ. Wow. So that's why it's very important that the percent similarity between the host and the recipient would be very high, so that we minimize the possibility of graft rejection. So what are the different forms of graft rejection? So graft rejections could be hyperacute. So when you say hyperacute, it occurs within minutes up to several hours after transplant. Hindi na nakatagal, no? So it is due to the fact that there have been preformed antibodies to ABO. By the way, if you're going to get an organ donor, it is very important that the recipient and the organ donor would have the same blood type. HLA endothelial antigens bind to vascular endothelium and active complement and clotting factors. So within minutes to up to several hours. Now, when you say accelerated, it occurs several days after the transplant. So the mechanism would also be the same as the hyperacute. Unlike in acute, which could last from days up to several months after the transplant, so acute is actually much longer as compared to hyperacute or accelerated. Okay, so here, the cytokine production by the T helper cell will induce delayed type hypersensitivity or the DTH. So the CD8 T cells mediate cytotoxicity. So the antibodies will bind to complement and will induce inflammation. And the same mechanism is also involved in chronic. It also involves the DTH. However, the rejection in the chronic could last for a year or more after the transplant. Okay? So, take care of your organs right now, because having an organ transplant is actually more complicated than you ever imagined. Okay, especially kidneys. Take care of your kidneys and other organs. Okay, so let's discuss naman, on the other hand, the graft-versus-host disease. Okay, the graft-versus-host disease. Okay, here, lymphoid cells in graft mount an immune response against recipients' histocompatibility antigens. Wow. So it's like a Trojan horse. Do you remember that legend? So when they received the horse, they didn't know that they have soldiers inside. So the same thing here in the graft-versus-host disease. Okay? When you get organs, for example, you receive stem cells, you receive lung or liver transplant from a donor, you didn't know that when they give you the donor, they also gave you the lymphocytes of the donor. And the lymphocytes of the donor present in that particular grafted organ will mount an immune response against recipient histocompatibility antigen. So it's like these lymphocytes act like a Trojan horse. So it involves massive cytokine release, inflammation, and destruction of various tissues such as the skin and the gastrointestinal tract. So it occurs 100 days or more after the transplant. Therefore, in order to prevent that graft rejection, some of these recipients or majority of these recipients would need to receive or or to undergo or to drink immunosuppressive drugs, such as corticosteroids. Corticosteroids is anti-inflammatory. Okay. Other. So what are the example of corticosteroids? Okay. So corticosteroids are potent anti-inflammatory and immunosuppressive. Okay. So, however, long-term use is associated with several complications, including hypertension and diabetes mellitus. Anti-metabolites will actually interfere with lymphocyte maturation and division. Okay? So, it will kill proliferating cells. So, example of anti-metabolite is azathioprine. Okay? And then the calcineurin inhibitors will block cytokine synthesis in T cells, such as cyclosporine and the FK506. FK506 is also known as tacrolimus. Okay, that will block the signal transduction in T lymphocytes, resulting in impaired synthesis of cytokines such as interleukin 2, interleukin 3, interleukin 4, and even gamma interferon. Okay? Monoclonal antibodies may also be utilized. It interferes with T cell or B cell function by binding to lymphocyte surface molecules. So, examples are basiliximab and daclizumab. Okay? So, both of these will bind to CD25. So, two anti-CD25 these drugs are the two anti-CD25 antibodies, which is of course important for the T cell and B cell function. Okay? And then the polyclonal antibodies are animal-derived antibodies against human cells that also deplete thymocytes and lymphocytes. Okay? So thymoglobin is an anti-thymocyte antibodies that were prepared in the rabbit, not prepared in the rabbit, and the other one is the ATGAM. Okay, so the other one is the ATGAM. So, what are the drawbacks of using immunosuppressive agents? Okay, the drawback is that it will make the recipient immunocompromised. Of course, the advantage that the possibility of you rejecting the newly transplanted grafted organ would be minimized, but not really 100% assured. But one thing is for sure, you will be immunocompromised. So you really have to take care of yourself and prevent opportunistic infection. I have actually a friend who recently received incompatible organ from her sister, and then eventually she got hospitalized in the ICU, and they found out that she developed pneumonia and the causative agent was Pseudomonas aeruginosa. So that's an example of being immunocompromised because of immunosuppressive drugs. Okay. HLA typing is actually the identification of HLA antigens or genes in transplant candidate or donor. So one of the most common or the classical way of doing this is by means of the complement-dependent cytotoxicity test. So that's the classic procedure in identifying HLA phenotype. Okay, so genotype is different because in genotype, it is a molecular method used to identify HLA allele. Okay, so the principle for the CDC or the complement-dependent cytotoxicity is that here you have panels of antisera or monoclonal antibodies, so predetermined monoclonal antibodies that define individual or groups of immunologically related HLA antigens. So they are being incubated together with the lymphocytes that you can see here. Okay, from the person to be HLA type in separate wells or a microtiter plate. So you have to screen. So you'll be harvesting the lymphocyte of So I think the specimen that you can utilize here is a buffy coat or the blood. Then you have to extract. You have to. They have this solution that will allow you to harvest the lymphocytes. Okay, so the lymphocytes of the donor, and it will be matched also with the lymphocytes of the recipients. Okay, so you have to screen. So the thing that you add here is actually the monoclonal antibodies. Okay, so they are being incubated. So each well, so you have several well, depending on how many panel that you are testing. So each well of the plate contains different antibody. So it is important to note that the multiple antisera are being utilized in HLA typing. Okay, so the requirement is based on the presence of both unique epitopes. Here, epitopes on HLA molecules, because the epitopes will define the phenotypic specificity of a certain HLA antigen. Okay, and there is also a public epitope. So you have a unique epitope and public epitope. So public epitopes are present on more than one unique HLA protein. So it becomes public. Okay, so because responses to public epitopes are common, many sera must be used to define pattern of reactivity that correlates with specific HLA antigen. Okay, so T and B lymphocytes are being used for class 1 typing. So this could either be T or B lymphocytes. So these are being used for class 1 typing, whereas purified B lymphocytes are used for HLA class 2 typing because class 2 antigens are not found on most T cells. Okay. After incubation with the antisera, we will be adding complement. So this is now the complement. So a complement reagent is added. So complement will eventually bind. Okay. Binding of antibody occurs only if the lymphocyte express HLA antigen targeted by the antisera. So you will be able to see here the antibodies in the antisera bind with the lymphocyte. Okay? A complement reagent derived from rabbit serum is added to each well, as you can see there. And if the cell possesses the HLA antigen defined by the antibody in that well, a complement is activated and the cells are killed. So the antibody bind here, the complement is activated, and eventually the cells are killed. So how would you know if the cells are killed? You have to utilize a dye. So you will be adding dye in the well, and that dye is actually the eosin red, or sometimes they are using tryptan blue. Okay, so eosin red or they are using tryptan blue. So we are using this to distinguish between live cells from dead cells that we can view microscopically. Okay, so the dead cells whose membranes have been more permeable are able to take dye. Therefore, all colored cells under the microscope are considered to be as dead. Okay, whereas the live cells membrane remain intact, which means they're still alive. Therefore, they cannot take up dye and remain colorless. So the proportion of dead cells is estimated by microscopic examination, and you have to score it according to the standard or scale that has been established by the American Society for Histocompatibility and Genetics. Okay, so grade 1, the score is 1 if there is 0 to 10% cell death, so it's negative. The score is 2 if 11 to 20% cell death, so it's doubtful, negative. The score is 4 if 21 to 50% cell death, so that's weak, positive. The score is 6 if there is 51 to 80% cell death, so that's positive. And then 8 if 81 to 100% positive. 81 to 100% cell death, that's strong positive, and 0 is unreadable. Okay, so it means that if the antisera that you use becomes positive, it means that lymphocyte is is actually positive for that particular HLA class. So now you have an idea what are the HLA classes of your donor, and then you have to compare it to the HLA class of your recipients so that you'll be able to know the percent compatibility. Okay. So, however, the more appropriate way of doing this is by means of HLA genotyping. Not anymore phenotyping, because in the previous test, that was an example of HLA genotyping. So here we are actually identifying alleles. So we are actually identifying alleles. So it has several advantages over CDC: higher resolution, reagent quality, and higher throughput. So it becomes more efficient in a way, but more expensive, I suppose. So HLA locus. So if this is the allele, okay, A is actually the locus because there are A, B, and C. And then the allele group is here, and 1 here is the allele number. And then this is the synonymous variation, and then this is the non-coding variation, and the letter N here is variation in expression. So there are several methods of doing this. So one of them is the PCR-SSOP, which stands for the sequence-specific PCR. Okay. So, and then we also have PCR-SSOP, the PCR, and the other one is the S. The PCR-SSOP stands for the sequence-specific oligonucleotide probe hybridization, and the SBT is the sequence-based typing. The most common approaches for analysis involve PCR amplification. So we know when you say PCR amplification, its principle is primarily DNA amplification. So we should be able to allow to amplify the DNA. So the HLA genes, say, with panels of primer pairs, so each of which amplified with I'm sorry for that. Okay, so where was I? So what I was saying that the most common approaches for analysis involve PCR amplification of HLA genes. Okay, so DNA amplification in particular. Okay, with panels of primer pairs. So in PCR, primer is indeed very important, each of which amplifies specific alleles or related allele group. Okay. So only those primer pairs that bind perfectly to the target gene result in detection of amplification product. So amplification is detected by agarose gel electrophoresis. So there will be several bands based on its molecular weight. Okay, the HLA genotype is then identified by determining which primer resulted in amplification. So for example, this is the principle of PCR SSP. So again, when you say SSP, this is the sequence-specific PCR. So the sequence-specific PCR, the sequence-specific primer encoding in 3' to 5' will be extended only from a template carrying the polyformism as shown here. Okay, so in this particular, this is the amplification control, and this is the amplified allele-specific product. In this case, there is no amplification. So that particular allele is not present in this patient case. So here's another one. This is the SSOP, or the sequence-specific oligonucleotide probe. So here, the patient DNA is amplified using primers labeled with either biotin or digoxygenine, biotin or digoxygenine. Okay. So actually labeled at the prime end. Okay, so the membrane or the amplicons. What is the amplicon? So amplicons are the amplified products. Okay, the amplicons are then hybridized. So when you say hybridized, combined to panels of probes. So probe is already the predetermined sequence, immobilized in a membrane. So this is the membrane. If the sequence of the amplicon matches the hybridized, nagmatch siya, kasi ito, amplicon may mga sequence to. And then this is the probe, may mga sequence to. Kapag nagmatch, then it will be hybridized to the probe. So a secondary reaction with enzyme conjugate, abitin or anti-digoxygenine will produce a color light. So kasi itong amplicon natin, meron na tayong avidin or digoxygenine. So mag-a-add lang tayo ng anti-digoxygenine, then there will be a light when exposed to substrate. So, if the sequence of the amplicon differs from that of the probe, syempre, katulad dito, ito yung probe, ito yung amplicon, hindi naman siya nag-match dito, so there would be no hybridization, and then there would be no color signal. Unlike here, nag-match sa probe, then there would be color signal. So pag nagkaroon ng signal, it means that the HLA gene or allele is actually present. Okay? We can also screen HLA antibodies and identify them. So HLA antibodies may develop in response to multiple blood transfusion, prior organ transplant, or kung meron ng history of patient, or paternally derived fetal antigens by pregnant women. So these antibodies can cause hyperacute graft rejection. So that's the reason why we need to screen and identify them. So donors with HLA antigens that can react with the antibodies in a transplant candidate should be eliminated from consideration. So eliminated na agad yung donor na to. Okay? So how do we test? So we can also test this by means of HLA typing by CDC method. Okay, baligtad naman. So kanina, we are testing the lymphocyte. This time naman, we are testing the serum. And baligtad naman, kanina, anti-sera yung ilalagay natin. But this time, we are adding panel of lymphocytes. So, anti-human IG can increase the test sensitivity. So, ang manner of reporting dito is yung percent PRA or panel reactive antibody. ELISA may also be utilized. It uses purified HLA antigen bound to microtiter state. So, ang testing for HLA antibodies, we are screening for antibodies. Hindi katulad ng CDC, we are screening for what MHC phenotypes are present on the surface of the lymphocyte. So, baligtad yun, guys. Okay. We can also do flow cytometry or cross-matching. So, after a suitable donor is identified for transplant candidate, so a donor-recipient CDC test is done to confirm the absence of donor-specific antibody. So, we studied flow cytometry before. So here, the patient serum is incubated with mixture of beads coated with different HLA proteins. So, our specimen here is serum. Then this is washed. And then we add the fluorescent isothiocyanate labeled anti-IgG. So, here, we wash and analyze for fluorescence on flow cytometer. Okay, so here is the particular HLA, and the HLA is actually coated in the beads. So, for example, the patient is positive for anti-HLA A11. So, the antibodies of the patient will bind with the bead. And then the anti-IgG-FITC label will bind at the Fc portion of the patient's antibody. And this is the one that will create signal, okay, in flow cytometry. Okay, so that's the end of our lesson here in Transplant Immunology. I hope you learned something. If you have any questions, feel free to message me, and I will also be adding supplemental videos coming from YouTube channels in this particular module so that you'll be able to understand the topic much better. Again, this has been Dr. Sipache Basit saying, stay safe and God bless everyone.